somewhat impermeable. It's not totally impermeable. But a less permeable layer on the outside will help you to live longer in this terrestrial environment. Yeah, that's good. That's a new development that comes into play. It gets very much more complex in the reptiles, ultimately turning into scales. You have a stratum corneum. This time of year, if you just take your fingernail and drag it down your arm, you'll see a white line left behind. Well, that's all of the dead cells of your stratum corneum that you just ripped off. Not all of them, but some of them. The epidermis in most vertebrates is interesting, not just because of what it does itself, but we have interesting things that are derived from the epidermis. They begin forming from epidermal cells. And very often, these structures are glands. That's true for mammals, for example. Mammary glands are derived from the epidermis. Scent glands are derived from the epidermis. Sweat glands are derived. All right, but we're talking about amphibians. And in amphibians, we're going to have two kinds of glands. We're going to have mucus glands and poison or granular glands. And let's say this is my epidermis with my dermis down here. What will occur is that cells will proliferate, cells from the epidermis. They start multiplying here and then push downwards into the dermis. So they originate in the epidermis. They're not a dermal structure, they're an epidermal structure. They came from these cells up here. So we're going to have some glands. If you look at them under the microscope, very often, most often, the major difference that you would see is that in one, it looks like there's a bunch of little granules. And in the other one, there isn't. So underneath the light microscope in a histological section of amphibian skin, it's very easy to identify two different types of glands, both of which developed from the epidermis. One appears granular because of the way it's stained, and the other one doesn't. The one that doesn't appear granular is the mucus gland. So mucus in amphibians is widely distributed across the body. Tends to have multiple functions. One, it's going to help keep the skin moist. Why do we want that? Because we need a moist layer on the outside of the animal to promote diffusion of oxygen across the body wall. Right, so it's going to help keep the skin moist. It might also be an anti-predator mechanism, and it certainly is in some. In other words, it makes the animal slippery, harder to attack. And it may be antimicrobial. In some, there's evidence that there are chemicals in the mucus that will help inhibit the growth of bacteria and fungi. If you think about it, that sort of makes sense. You've got dead skin on the outside. Bacteria and fungi should be happy to consume that. But you don't want to walk around with all of these colonies of bacteria and fungi on you. So an antimicrobial function might be useful. Okay? The granular glands are, to me, more interesting because they do produce a toxin, a poison. And these kinds of glands are present in all amphibians. So in our nice little leopard frogs and wood frogs, they have poison glands inside their epidermis, right, derived from their epidermis. These glands may be clumped in particular areas, like the parotoid glands and the warts of toads, or they can be broadly distributed across the skin, as they are in ranid frogs, for example. What does the poison do? Well, broadly speaking, it's either going to have noxious effects or toxic effects. With noxious, that's probably the most widespread effect of the granular glands of amphibians. It gives maybe the animal a bad taste or a bad odor, or perhaps it causes the animal to vomit, a predator to vomit if it takes that into its mouth. I don't know if you've ever seen a dog after it has tried to eat a toad. They can be foaming at the mouth and they're just not happy whatsoever because they clamped down onto that toad and released some of this material from the parotoid glands and elsewhere on the body, and it's all in the mouth, and they will potentially throw up. I've seen snakes throw up after eating toads. Right? Sometimes the toad's alive, sometimes it isn't. But that's the most widespread type of poison in these poison glands. Some of them, though, are downright toxic, potentially deadly. They have a variety of effects. Some can produce convulsions, hallucinations, heart arrhythmias, paralysis, and of course death. The most potent of these toxins is found in the family Dendrobatidae. These are poison arrow frogs. Poison dart frogs is another name that you hear them called. They're found down in the neotropics, so starting in southern Mexico and then down into South America. But there's one species, just to illustrate, Phyllobates. I love the name, that's why I'm putting it. Phyllobates terribilis. So, inside its poison glands, there's an alkaloid, and it increases cell permeability to sodium. Because of that, this material causes depolarization of muscles and nerves. So the nerves cannot transmit impulses, and the muscles cannot relax. So pretty much you've got paralysis of the animal, and you end up with heart failure and death. The natives down in the indigenous people in those areas would take these frogs, heat them up over a fire, which ticked off the frogs, so they exude their poison. And then they would put that on their spear tips or arrow tips or dart tips, and they'd go hunting with it. Right? Because then it would go into something like a little deer and cause paralysis, and it would be much easier to hunt. All right, that's at one extreme. Also, remember that I didn't mention it to you guys. Colorado river toad out east, out west, is an endangered species, and one of the reasons it's endangered is because people would have the habit of going and picking it up and licking it, because the toxin would cause hallucinations. Going along with extreme toxicity, we might as well mention aposematic coloration. Most of the very, very toxic ones are aposematic. In other words, they are brightly colored. And this is warning coloration. So aposematic coloration is warning coloration. In other words, instead of being secretive and brown or secretive and green, so you're hidden in the vegetation, these guys are bright blue, yellow, red, orange, whatever. And that is a signal then to potential predators. Don't mess with me, because if you do, you're going to be sorry. Right? And we'll see aposematic coloration in other groups when we get to the reptiles, for example. So it's something that occurs in numerous groups of organisms. Any questions on that so far? Pretty straightforward.
I have one about the tympanums.
Yeah.
So they're saying that females have a smaller tympanum than males. So
Interesting.
I read that.
So we may hear that.
Is it, I know some species that's true. Is that a consistent?
No, it's a lot of some species, and they're saying that the females have a smaller tympanum. They said that some species, the females have the smaller tympanum.
Yeah, and I know that's true, and I know where you're going. It's an interesting point. Anybody else know where he's going?
Does that,
Well, go ahead.
oh, well, does that mean that they hear the sound differently compared to what the noise that the males are actually making?
You know, just straight from the size of the tympanum, you would think.
That's, well, what matters isn't the size of the tympanum, it's the ratio of the size of your tympanum to the size of the window that it goes to. So we have about 20 times amplification to the size of our eardrum versus the size of our open window. So,
Yeah, you would have to look at it in more detail than just the outside. So if you're just looking at the outside, you would think that potentially a female can't hear as well as a male. But he's right, it's the matching that really, really counts. And intuitively, you would think it should be the other way because we're just looking at the tympanum. And the whole idea is that female needs to hear the male. Right? But just because the tympanum itself is slightly smaller in dimensions doesn't mean that she has poorer hearing. Okay. Let's start on metamorphosis. Metamorphosis. All right. We can define metamorphosis then as a series of changes involving structural, physiological, biochemical, and behavioral changes. Right, so it's a series of changes, usually somewhat abrupt in the scheme of the world, involving structural, physiological, biochemical, and behavioral transformations. We're going to be changing something about the structure, changing something about the physiology, biochemistry, or the behavior. And these changes take place over a reasonably short period of time within the animal's lifespan. We can categorize these changes, whether it's structural, physiological, biochemical, behavioral change. We can categorize the changes into 3 categories. And those categories are regression, transformation, and developments. So we have changes in all of these things: structure, physiology, biochemistry, chemistry, behavior. We can take each one of those and classify it as a regression, a transformation, or a development. What do I mean? Regression then involves structures and functions that are needed only by the larvae. So these structures or functions are going to disappear. They're going to regress because they're needed by the larvae, but they're not needed by the adult. What would be a good example in an amphibian? A tadpole versus an adult.
Gills.
Gills, that's a real obvious one. Okay? You have an aquatic larvae. It's living in the water. It's using its gills in order to obtain its oxygen, but it metamorphoses when it goes out onto land because gills are not useful on land. So the gills regress. A tail in frogs. All tadpoles, frog and toad tadpoles, have a tail when they're in the water, but that regresses because it has no use for them when they're on land. Okay, so those are regressions, things needed by the larva, but not needed by the adult.